So the last thing that we want to talk about is going to be acids and bases. Now the interesting thing about water is that water itself can ionize. Ionization is when we have a single molecule that is held together that breaks down into individual ions. So for example, sodium chloride or NaCl can break down into sodium ions and chloride ions when mixed into water. The weird thing about water is that when water is mixed into water, or basically when you have a cup of water, it breaks down and ionizes itself. It's going to break down into positively charged hydrogen ions and negatively charged hydroxide ions. But here's the weird thing. It breaks down at the same rate that they come back together. Those positively charged hydrogen ions are attracted to those negatively charged hydroxide ions. So they are going to be breaking up and getting back together and breaking up and getting back together and breaking up and getting back together more times than you can even imagine. So water is going to dissociate and ionize and release an equal number of hydrogen and hydroxide ions. Now the funny thing about this is that this is directly related to pH. pH, you may have heard before, is how we measure the acidity or the alkalinity of a solution, and really all that is a measure of the hydrogen ions that are present. So we're going to talk a little bit about this. Acidic solutions are going to release a lot of hydrogen ions when they are dissociating in water. So when we put acidic solutions in water, they're going to break up and they're going to start sending a whole bunch of hydrogen ions out there. Things like lemon juice, tomatoes, coffee, they all dissociate in water and release hydrogen ions. The acidity of a substance depends on how fully it dissociates in water. Strong acids are going to break apart completely in water. Weak acids are only going to dissociate partially in water. Basic solutions, on the other hand, either take up hydrogen ions or they release hydroxide ions. If a strong base is placed in water, the number of hydroxide ions is going to increase. Those strong bases are going to dissociate fully in water. So what happens with an acid is going to release a whole bunch of hydrogen ions. A base is going to release a whole bunch of hydroxide ions. Water, on the other hand, releases a whole bunch of hydrogen ions and a whole bunch of hydroxide ions, and it does it in the exact same equal concentrations. This means that that water dissociates in itself to release just as much acidic as it is basic. This is why water is considered to be neutral on the pH scale. The pH scale is used to indicate the acidity or the alkalinity of a solution, and pH scale measures the concentration of hydrogen ions in a solution. The pH scale ranges from 0 to 14, with 7 representing neutrality. So 7 is where we have just as many hydrogen ions as we have hydroxide ions, meaning that it is just as acidic as it is also basic. This is where water lies. The pH scale is also logarithmic. It means that each unit change represents a 10-fold change in the hydrogen ion concentration. So as you move down the pH scale from 14 to 0, each unit is 10 times more acidic than the previous unit. So if pH 7 is 10 times more acidic than pH 8, pH 6 is 10 times more acidic than pH 7. So how much more acidic is pH 6 than pH 8? So if there's a 10-fold increase in acidity from 8 to 7 and a 10-fold increase from 7 to 6, that means there's a 100-fold increase between 8 and 6, meaning that pH 6 is 100 times more acidic than a pH of 8. So it's very important that organisms are able to maintain standard pH in order to maintain their homeostasis, because if we change a little bit on the pH scale, it's actually a really drastic change in hydrogen ion concentration. It can throw everything off. So the way we help maintain, just like we need to maintain a constant internal temperature, we also need to maintain a constant internal pH environment. We're going to do this by using a buffer. A buffer is going to be a chemical or a combination of chemicals that keep pH within a narrow range of limits. In living organisms, pH of body fluids usually needs to be maintained with a very narrow range in order for the molecules to function correctly. If we change pH too drastically, we're going to see this when we do our lab on enzymes, but if we change the pH too drastically, we can completely destroy the functionality of those proteins. Buffers help us maintain those normal limits by either taking up those excess hydrogen ions or the excess hydroxide ions. They basically react as a sponge to soak up all the excess. So for example, in a human being, human blood is normally around a pH of 7.4, ever so slightly basic. If that pH drops below 7, acidosis results, and if the pH rises above 7.8, alkalosis results. Both of these can be life-threatening situations. So what we have in our bloodstream is going to be what's known as carbonic acid, H2CO3. It's a weak acid that minimally dissociates and then reforms. The dissociation of carbonic acid leads to the release of hydrogen and bicarbonate ions. When those hydrogen ions are added to the blood, they can be absorbed by the bicarbonate ion to form carbonic acid. And when hydroxide ions are added to the blood, they can be absorbed by the carbonic acid to form the bicarbonate ion in water. So that reaction is going to reduce the basicity. So basically it sits there, and if the scale ever tips too far in one direction, it's going to soak up the excess. So that's it for this chapter. I'm going to just do a quick summary. We first talked about chemical elements. We talked about atoms and how they contain subatomic particles. They contain protons, neutrons, and electrons. For each of those, I would like you to know where they are located, what their charge is, and what their mass is. So for protons, neutrons, and electrons, you should know where they are located, how much they weigh, and what their charge is. You should also know that isotopes differ in their number of neutrons. If we start changing the number of protons, we're going to completely change the element itself. But if we change the neutrons, we can change its stability and we can form isotopes. Electrons are going to be found in the electron shells orbiting around the outside of that nucleus. The valence shell is going to be the furthest shell from the nucleus, and it's going to ultimately determine the reactivity. You should also be aware of the octet rule that states that the atom is going to be most stable and most happy when it has a completed valence shell. If that is only the first energy shell, we only need to have 2 electrons in it. If it is any shell outside of that, it needs to have 8 electrons in order to be completely satisfied. Then we moved on into molecules and compounds. A molecule is going to be 2 or more atoms. A compound is going to have 2 or more elements. We talked about chemical bonds. We talked about ionic bonds and how that's an interaction of oppositely charged ions that are brought about by the donation or acceptance of an extra electron. Then we talked about covalent bonding and how that's formed by the sharing of electrons. However, sharing isn't always equal. When we do have an equal sharing of electrons, we form what is known as a nonpolar bond. However, when we do not share equally, when one atom is spending more time hanging out with those electrons than the other, we form what is known as a polar bond. This is an unequal sharing of electrons. This is going to create a partial charge at one end of the atom, or one end of the molecule. And we talked about the chemistry of water. We talked about hydrogen bonds. Make sure you are familiar with those. They're going to come back again and again and again. So be familiar with hydrogen bonds, how they're formed and what they do. And then we talked about the properties of water. We talked about its high heat capacity, high heat of vaporization, how it functions as a solvent. It's both cohesive and adhesive, and it's less dense in its solid form. Finally, we finished out with acids and bases. We talked about how water molecules will disassociate when they are sitting in water, and then we talked about how the pH indicates acidity or the alkalinity, the acidicness or the basicness of a solution. This is just going to be a measurement of the hydrogen ion concentration. Acids have a pH less than 7, bases have a pH greater than 7, and buffers are what we use to help maintain our normal physiological pH. That's it for this chapter. If you have any questions at all, please send those to me in an email, and I will be sure to answer them in our next discussion meeting.